Like most of you, I know what it's like to be in the lab late at night and all you want to do is fix your samples so you can go home and run them the next day. Fixatives are fantastic for that. They make your experiments very convenient, they allow storage of your cells, and they're going to prevent your samples from becoming infected. However, there are also some negative side effects to using fixatives, like increased autofluorescence or changes in scatter. So it's important to think about the type of fixative you're going to use, how it will be applied to your experiment, and what effect that fixative may have on your fluorescence detection. There are two main classes of fixatives: alcohols and aldehydes. So talking about alcohol fixatives, this is going to be your ethanol and methanol. When you look at protocols, you'll generally see these used with things like cell cycle analysis. And that's because these fixatives, alcohol-based fixatives, are really great for fixing DNA. However, they can be very poor at fixing proteins, and this has to do with how they work. So as your alcohol fixes the cells, it denatures and precipitates the proteins. This may hide antigens, so they're no longer recognizable to be bound by antibodies, but it also can destroy the structure of protein-based fluorochromes, so you may actually lose fluorescence in your dyes when you add these fixatives. So like anything else, it's really important to test them in your experiment to make sure that they are going to work. Now, some pros of alcohol-based fixatives. Once your cells are fixed in ethanol or methanol, they can be stored at -20 indefinitely. You can go back to them months later and your cells will still work the same as they did the day you fixed them. Another pro is the alcohol will permeabilize your cells because it dissolves the lipid membrane. So when you use an alcohol-based fixative, you don't require any subsequent permeabilization steps to stain intracellular targets. The most important thing to keep in mind though is that not all of your fluorochromes can withstand alcohol-based fixatives. So if you're going to do a surface stain and then fix with alcohol, it's really critical that you test to make sure that your fluorescence is maintained after your cells are fixed. So the second class of fixatives are the aldehyde-based fixatives, and you'll probably see these used much more commonly because they work better with proteins. Now let's start by talking about paraformaldehyde. Paraformaldehyde, oh boy. So, I would like all of you to stop referring to your fixative as paraformaldehyde. Paraformaldehyde is a white powder. It is not fixing your cells. As soon as it's solubilized in liquid, your cells are fixed with formaldehyde. So even though you might be making a 4% solution from paraformaldehyde, as soon as it is diluted and resuspended, it is now formaldehyde. So please stop referring to it as paraformaldehyde in your paper. Aldehyde fixatives work by cross-linking lysine residues, so basically cross-linking your proteins together and locking them in place. Because of this, you don't have as many structural changes as you have with ethanol, and so more of your protein structures will be maintained, allowing you to stain sometimes after you've fixed. Again, you'll need to test this with your specific antibody-antigen combination. However, generally speaking, aldehyde fixatives are good for proteins, bad for DNA. Generally, formaldehyde fixatives are used at about a 0.5 to 4% concentration. So like everything else, you will need to test to find the appropriate concentration for your experiment. I know I've made this mistake in the past, and I've left my cells in fixative overnight. And while the cells still looked more or less the same, what you have happen is an increase in autofluorescence. So the longer your cells are in this fixative, the higher your autofluorescence or your background spread gets, making it more and more challenging to separate your negative from your positive. So always keep in mind that 10 to 30 minutes is more than enough time to fix your cells. Anything more than 10 to 30 minutes and you're going to start reducing your ability to distinguish your negatives from your positives. And especially if you're looking at dim populations, this could make or break your experiment. Another important note to aldehyde fixation is it is an exothermic reaction, meaning that it produces heat as your cells get fixed. Because of this, you always want to do this fixation cold. I recommend on ice. In the fridge also works. Another tip with formaldehyde fixatives is the fixative is going to fix your cells in whatever state it finds your cells. So if you add fixative to your cells when they're already clumped up, you will never break these clumps apart because your cells will be fixed in clumps. So it's very important to have your cells thoroughly resuspended in a single cell solution And if you can, add the fixative dropwise whilst continuously mixing your cells so that they don't clump together while you're fixing. So to quickly recap the aldehydes, again, working with formaldehyde, not paraformaldehyde. Keep your concentration as low as possible, but make sure you've tested it. Test the amount of time required, but try to keep it under 2 hours. Always fix on ice, and make sure your cells are single cells so you don't fix clumps together. And doing those things, aldehydes can be very much an important part of your flow cytometry buffer repertoire.